today's main theme will be related to the reaction-diffusion systems. I'll talk about the mathematical analysis and my numerical simulations. And there will be two important concepts today. I will be talking, one is the geometry, and the other is the effect of cross-diffusion in reaction-diffusion systems, especially to the pattern formation observed in the nature. Okay. So this talk is actually extracted or summarized in a way that taken from the three papers here that you can see. This QR code is linked to the published work. The first one has recently published, and I will try to give you my observations, my findings, my results related to these three papers. So this is the outline. I will be giving a brief introduction, giving the biological motivation, why we are doing this, why we are working on, and then I'll show you the mathematical model, and then I'll be talking about for our model to understand the patterning dynamics, we need the eigenvalue problem and its solution. I'll talk about that, and then we will arrive at stability matrix, which is necessary to understand spatial pattern formation and spatiotemporal pattern formation. And now with the help of our analysis in 5, we can able to generate our parameter spaces. And then I will show you my numerical simulations obtained by finite element method. I'm not going to go into details of finite element method. Rather, we use the finite element method here to verify our theoretical findings. Then I'll show you my conclusion and what I'm working at the moment and what I plan to do in the near future. Okay, it is originally Alan Turing theory. Alan Turing is generally he's known by his works about cryptography. He's a mathematician, but he published a paper in 1952 in chemistry, titled with the chemical basis of morphogenesis, and he tried to answer the questions like how the animals get stripes and spots on their body. And he tried to explain, he explained like the pattern formation in nature is because of the responsibility of reaction, some sort of reaction and diffusion. So I'll talk about the Turing's theory a little bit more, but before I want to talk about what kind of patterns that I work and what is a pattern formation in nature. I'll try to show you. Okay, here we have this nice two figures saying that the first one is a string ray, and we can see like how the spots occur on its body, and there is spots type of patterns and there are stripe type of patterns, as you can see in the second one in zebra. So we can either have some spots or stripes, and it's not limited to these kind of patterns. We can see other kind of patterns like different kind of waves. This is what I mean. So as I told you, today the main theme is going to be the geometry and the cross-diffusion, but our analysis is depending on the domain size. So why domain size is important? Because in the embryonic development, so we know that during the growth development, it reached some certain sizes so that you can have these kind of patterns in nature. So, for instance, you have a fish, and this kind of fish that you are feeding, and at some time later you see that your fish has evolved and it has some more stripes, more patterning on it, and finally as it's mature at its reach at some level, you will see like you will have like lots of elegant patterns on it. So here I just wanted to show you like the growth, the importance of the domain size here. So therefore it's gonna be entitled to my work, why we are working on domain growth, because the domain growth issue is quite an important subject in reaction diffusion systems. Another example I want to give you from nature is the limb development, digit development. For instance, I think everyone can see my hands at the moment. So during the growth development embryonic stages, so before our fingers has evolved to this natural sizes, so we see this type of shape, and you see the snapshots from the figures that once the evolution happens, our fingers are revolved and we have this shape in a healthy hand, 5 fingers. And the like for the lifetime for the long-term dynamics, our finger size is same pretty much, as long as we are talking about the healthy circumstances. And these are all the examples of spatial kind of patterning. So we have some space variations, but they are all stationary in time. It's like the limb development, for instance. So there are other kind of patterns that we can observe in the nature. It's a lot actually, a lot common. When we have the temporal periodicity in the dynamics. For instance, so I would like to go back to this first string ray figure. It actually has some special properties that, according to the environment, it has the ability to adapt itself. It changes its patterns. The patterns are disappearing, are evolving, and then they come back to the same topology again. So it's like there is a cyclic structure here. From the spatiotemporal analysis, what I mean to say that there's a cyclic structure, like our hearts, like heartbeat, like we breathe and we exhale and inhale, and there's a cyclic structure. I will be talking about this cyclic structure as well. Okay, a little bit more on math now. This was my introduction. Feel free to interrupt me if you have any questions. Reaction-diffusion systems, they are given by mathematical models. They are a set of equations that they include the reaction and the diffusion terms. I will show you, and I want to talk about a little bit more on diffusion, the meaning of diffusion and the cases of diffusion, because specifically this work is motivated by the effect of cross-diffusion. That's why I would like to explain it. So the idea of Turing diffusion-driven instability, it says that you have a set of reactions. Think about that. You have a system with reactions in the absence of diffusion, where you have a homogeneous, linearly stable, like your steady state is linearly stable. But then diffusion is added to the system, and then your system becomes unstable. So it is called as a diffusion-driven instability, and this is what we are interested in. I'll show you some instability and how instability occurs. But this is what Turing theory suggests that. So the diffusion can be characterized in several forms, such as self-diffusion. It could be like including 2 components or more, or cross-diffusion. What is the definition of cross-diffusion? It's defined as it's a process. The gradient of one concentration of one chemical or biological species indexes the fluxes of the other. Like the diffusion rates are really codependent on each other. Okay. The importance of linear cross-diffusion, I want to talk about it. It has like a lot of applications. Is in solitons, especially in material science, like to understand the interaction between molecules, proteins, salts, different kind of biological or chemical species that are interacting with each other, and with different cytos especially. And it has also has a lot of applications in animal ecology, like the animal behavior, predator-prey systems, especially chemotaxis. And it has really significant and positive effects on the ability of reaction diffusion systems to form patterns. Okay, one more observation I want to talk about the Turing systems is that long-range inhibition and short-range activation. Suppose that you have 2 components, the activator and inhibitor. According to the Turing's theory, it has been proved both experimentally and mathematically that the diffusion rate of the inhibition must be a lot larger than the diffusion rate of the activator. Therefore, we can have the patterns in nature as it is. So it actually tells us that they cannot have equal diffusion rates. The inhibitor must diffuse much faster than the activator. That's what Turing theory suggests for the reaction diffusion systems without cross diffusion. I want to take attention to this point once again, that by adding cross diffusion to the system, now this condition has been relaxed. We now have ability to have the diffusion rates to be equal to each other, and it has again been proved in experimentally or also mathematically. So here are some examples without reactions. We see 3 2x2 matrices. These are the diffusion matrices, all of them. Our self-diffusion coefficients stay on the diagonal entries, whereas our cross-diffusion coefficients are staying on the off-diagonal entries. If you look at the first experiment, cell-cell solution, we have some reasonable rates of like compared to each other. I'll tell you the relationship that's between these four coefficients here in a little bit later with some mathematical form. So what I just wanted to show it here, that I hope that you can see my cursor. We can see the cross diffusion as a very small or a very large rate, or even negative. Negative cross diffusion is also possible. Negative cross diffusion is referred to the idea that we have some attraction to the size of the one molecule, like from the bigger one to the smaller one, or vice versa. It affects the sign of the cross diffusion. But our self-diffusion coefficients are, of course, never negative. Okay, what are my contributions to this to the literature? I would like to talk about it. So it is the relationship between the domain size and the parameter values. It means that how large a domain should be, therefore we can choose our parameter values, therefore we can have the pattern formation with respect to the domain size. And in adding to the Turing instabilities, the one that I'm talking about, I'll be also talking about a Hopf or transcritical type of bifurcation, referring to the spatiotemporal patterning. Like we have the spatial variations, also temporal periodicity. So I'll show you those kind of bifurcation results as well. So then I have several conditions. These are newly suggested conditions that it is actually gives us an understanding in parameter spaces. Then, of course, we are doing all the analysis in addition to the cross diffusion to the system. And I will show you why parameter choice is important, because think about that. You have a system, and parameter choice is of course very important, because if you randomly choose your parameter, unless you are very lucky, it's not going to be an effective way to simulate your work. You might have your system like abrupt patterns, abrupt solutions, or even no solutions, no patterns. And I say no solutions, I didn't mean say non-existence of the solution, no patterns at all. That's why by doing the analysis, we know how we can choose our parameters. So therefore we use the finite element method, and I will end my talk. So here is the mathematical model. When you look at that, it's consisting of 2 equations, namely the U and V. They are the components. U is the activator, V is the inhibitor. So a reaction to happen, we need an inhibition. We need an activation and inhibition. And I hope you can still see my cursor. This is the dimensional form. This is the non-dimensional form, sorry. This is the non-dimensionalized form of the system. And this D here shows the main diffusion coefficient, self-diffusion coefficient. This dV and du here are our cross-diffusion parameters. And this F and G, they are the reaction term. Gamma is our reaction scaling parameters. And this system is analyzed under zero flux boundary condition, Newman boundary condition. So this D, because this is the non-dimensionalized form, during non-dimensionalization, we say that this D is the ratio of the diffusion coefficients dV and du. Here, the long-range inhibition actually refers to the idea that this dV must be a lot larger than this du, referring that this ratio should be greater than 1 in Turing systems without cross-diffusion. But now with adding cross-diffusion to the system, now we can able to choose those coefficients are equal to each other, and we still have some patterns. This is one positive effect of the cross-diffusion. Okay, du and dv are our d, du, dv, and gamma are main parameters in this system. Okay, well, closeness of the PDE, the relationship between the main diffusion and the cross-diffusion parameters should follow this relationship for the PDE to create positive solutions. It has been shown in this paper, and our parameter choice and our analysis is all done at first with this assumption, the relationship between the diffusion parameters. Next, the second theme of my work is the geometry. So I worked on two-dimensional geometries, but I start with a rectangular domain. So I was interested to see what happens on circular shapes. Disc-shaped domain. I'll show you three different geometry here: rectangular, disc-shaped, and ring-shaped domain. So ring-shaped domain here is our non-convex. Think about an annulus. Like we have the outer circle and outer disc. Sorry, we have an annular region. How shall I explain? Like we have the outer, think about it, outer circle and inner circle, like the thickness is b minus a. So this L in the rectangular region is going to show the domain size, because we need to relate the domain size with the system parameters. That's why I would like to introduce you with the domain size parameters, controlling parameters. Domain size controlling parameters for a rectangular geometry is L, and this rho is the domain size of the disk, and b squared minus, b minus a is going to be, we are going to call it as rho. B minus a is going to be the thickness of the annulus. Okay, so another important concept in reaction diffusion systems is to define the reaction kinetics. There are a lot of reaction kinetics explained in the literature. Here in our work, we use the Schneckenberg model, activated depleted model, it's called. It has, again, these are also after non-dimensionalization process, you can see F and G. And this is good that Schneckenberg model is known to have the uniform steady state, which is good, because in many reaction diffusion systems with some more complicated reactions, especially in cell migration, in cell signaling, there are a lot of reaction kinetics which is really difficult, quite difficult, or even non-exist to find a unique steady state. So our Schneckenberg model has a unique steady states, and how do we do the analysis? The analysis is the linear analysis, linear stability analysis. It is done around the uniform steady state, and we start the procedure by linearizing the nonlinear reaction terms by using the Taylor expansion and around uniform steady state. So therefore, we have the linearized matrix vector form in a compact form, you can see. And where we have the W as the solution vector, we are interested in finding U and V diffusion matrix. I showed you the experiment in the beginning about this. And G is the Jacobian matrix. And those values are calculated at the uniform steady state. And the second part of the linear stability analysis is to work on the eigenvalue problem. Because eigenvalue problem is finally going to give us the relationship between the geometry, between the size actually. So on each geometry we have this eigenvalue problem under Newman boundary conditions. Okay, so we approach the solution of this eigenvalue problem for the rectangular region, for disc-shaped region, and for annular region. As you know that for the disc-shaped domain and annular domain, there is some harmonical analysis is needed. I'll skip those analysis, but you can anytime go to, I can refer you to these papers. And okay, the important parts of this is like we have the first one as the eigenfunctions, and the K-squares are representing the eigenmodes, eigenvalues. Here, L, as you can see, appear in the eigenmodes. So because we need this L to relate with our system parameters. Therefore, the solution of the eigenvalue problem is going to be quite important in the analysis. And in the disk-shaped domain, it will, because of the geometry, it will require using the radial and angular functions and how shall I say, solution of the Bessel differential equation give rise to the eigenfunctions. Eigenfunctions are going to be like in the form of as a multiplication of radial and angular functions. And as the geometry gets, how shall I say, gets, I don't want to say complex, but advanced, let's say, from rectangular to disk, as you can see, our eigenvalues having this form, it has more terms in it. And here, for the disk shape, for rectangular domain, it's all that which is important. For the disk shape domain, it's rho that this is the domain weight. And for the annular domain, it's going to be rho. It's the outer circle is somehow embedded in the simplification. Therefore, you don't see B minus A. Instead of B minus A, you see rho here. Rho is going to be here to show the thickness of the annulus. Okay, the solution of the what are we doing with the solution of the eigenvalue problem? Actually, we use them in our system, and therefore our solution is written by using separation of variables as a product of spectrum of the Laplace operator and time decay, exponential decay. And therefore, we are going to have this on the rectangular domain. Actually, for the two other domains, it's a similar approach, but I put the results in the end, because at the moment, this is what we need to use in the system. So we use it in the system. Therefore, we are going to have fully linearized dynamical system of ODE, having this time, as you can see, k squared, gamma, d, du, dv. All we need is now inside the matrix on the right side. And when we calculate f, g, remember those are the reaction functions, and we calculate their partial derivatives, and we substitute into the system, and it will lead to the two-dimensional discrete eigenvalue problem, algebraic discrete eigenvalue problem. And the solution of this problem, actually, this matrix on the right is called as the stability matrix, is referred to the stability matrix. And solving this equation requires finding the roots of the characteristic polynomial in quadratic equation in lambda. And we can, based on the observations, we can write the roots of this characteristic polynomial by using the trace and the determinant of the stability matrix. This trace, the expansion of this trace, these two things are here important, because when you look at the trace of the stability matrix, it includes only one diffusion coefficient. It's the main diffusion. Therefore, when you look at the determinant of the stability matrix, it includes everything. It includes the domain weights. Domain weights are embedded into k squared. I mean, if I prefer to write it explicitly, it's going to be quite large. Remember the expansions of those k squares. I just let them implicitly here. So our determinant includes all our parameters d, du, and dv. Okay, the rest is going to be done by using the sign of the eigenvalue. So we are going to see our eigenvalues. The choice of eigenvalues is going to give us information about the stable and unstable regions. Okay, since we are interested in spatiotemporal analysis, we are interested in considering the complex conjugate eigenvalues. Therefore, we need to say that it's actually the curve that is going to given by 19 is going to separate the real and complex eigenvalues, but we want our analysis to be done on complex pairs. This 20 therefore is going to give us that t square being smaller than a quantity. That quantity is a direct requirement of determinant to be positive. That's a good observation, because our analysis starts with analyzing the trace, but trace doesn't include the cross-diffusion parameters. It only includes B. but our determinant includes all the system parameters. Okay, here is the first observation. This is done by considering the real part of the root. So we want our real part to be the positive, real parts to be positive, which means that trace is positive. And trace being positive give rise to the condition S21. And one attention about this condition is that it tells us that domain size controlling parameter must be large enough to satisfy this condition. It means that this condition must be fulfilled for the system to exhibit Hopf or transcritical type of bifurcation. So we are going to say L must be large enough to satisfy. And again, this doesn't include any cross-diffusion parameters. Therefore, it's going to stay in the same way without the cross-diffusion. The work actually is an extension of this work done by, these are my collaborations in this paper. I'll mention about them in the end. So this is what I want to say is this is true for without cross-diffusion. And in the second theorem here, it forbids the other side of the, for L being less than the right-hand side. This actually tells us it forbids the transcritical bifurcation, Hopf or transcritical type of bifurcation. It only allows Turing type of instability. It only gives us the spatial variations. It doesn't give us that temporal periodicity. It gives us temporal stationary in time solutions we are gonna have. But I mean, there is no cross diffusion in it. So we really want to include our cross diffusion parameters d, du, and dv, all of them together. We want to see the effect of cross diffusion into our analysis. Therefore, we derive the conditions by using the positivity of the determinant of the stability matrix. So I want to go back to the determinant. This is the determinant, and we are interested in finding the positive solution of this expression. And then this will, for each kind of geometry, it has different conclusions, of course. So therefore, we arrive at a condition that L, domain size, must be large enough to satisfy the right-hand side. The weight on the right-hand side should be smaller than the domain side. So we need to pick our parameters based on this assumption. It is important. So it is done for each of the geometry, and since we have the domain weights in our eigenmodes, those eigenmodes are actually some parts of it, they are with the system parameters on the right-hand side of each inequality. So this is for the disc-shaped domain, this is for the ring-shaped domain. As you can see, as the geometry gets advanced, we have the conditions look like a bit like a tricky way. Okay, to understand what this condition says, a numerical representation is needed. Based on the assumption, I created this full classification figure. According to this figure, it tells us that, it really tells us from which region, if you choose your parameters, you will have patterns, spatial patterns or spatiotemporal patterns, or even no patterns. For instance, if you pick your parameters from A, where you have the real distinct negative eigenvalues, there will be no patterns at all. Or in B, the difference between A and B, that's those curve, remember, this is the curve that, there is no curve actually here. This is like color coding region, like saying that we have the complex pair with positive real part, we have the real parts, we have the complex and real parts, simply. The magenta region is the one where you have the real negative eigenvalues. The green region we have the complex eigenvalues, but has negative real part, which we are interested in the positive eigenvalues. Therefore, we need to go deeper into the regions C and D. The difference between C and D that we know now that from the C region, from region C, okay, from region C, makes me a bit distracted, sorry. Okay, from region C, we are gonna have spatial temporal patterns. We have spatial variations with time periodicity. And from the region D, we are expecting to have only, we know that we are gonna find Turing type of patterns only. Okay, the detailed, a larger version of these parameter spaces are here. And now we want to vary system parameters because we really want to know which system parameters are good for us. When I say good, we want to have some patterns. That's why we want to vary the system parameters. So we vary the system parameters based on the assumption that our domain is stationary. We fixed our domain, let's say for the rectangle domain, we fixed L to be 1. But these regions are actually based on the from the disk-shaped domain, those regions. I didn't want to, I'm not going to show all of the regions because they have the similar idea. So we vary the system parameters, and we can see the limit cycle curve. We can see how where we can have the Hopf bifurcation type of solutions. And these are the Turing regions, those regions that is the larger version of D here. So again, we vary, we do variations of parameters like we vary D, du, dv, and gamma, and we see how our regions are enlarging or shrinking, like getting larger or smaller. We see such observation. Okay, now, is there any questions so far? Because I'm now going to show you my numerical simulations obtained by finite element method. For the Turing type of behavior, I didn't generate a video, but I generated a video for you to see how spatiotemporal patterning occurs in the dynamics. Okay, the Turing instability, like from the beginning, we say the evolution happens, but once the evolution happens, the patterns stay as it is. There is no time evolution. It's stable in time, as you can see, like limb development, like patterning on an element, animals. Okay. Another simulation is now done for the disk-shaped domain. Now you can see how it's evolved from the snapshots. The evolution is completed until the time level not exactly, approximately the time level of 0.5. We have the patterns, and the pattern stays as it is in the long-term dynamics. They don't change. We run the simulation for even for larger, but we can see from the L2 norm graph that the solutions are converging. We have converging to inhomogeneous solutions. So therefore, the L2 norm graphs actually show the convergence of the solutions in numerical method. One good thing is that I mentioned in the beginning, the addition of cross-diffusion has the ability to perform the pattern formation. Without cross-diffusion, such simulation doesn't exist, because the choice of the main coefficients being 1 is not allowed in classical reaction-diffusion system. By adding cross-diffusion to the system, we are now able to generate those patterns. Again, it's a time temporally stable spatial variations we see. We have space periodicity. Okay, now this is the behavior of a spatiotemporal pattern. According to the idea, there is a change in time. It tells us that the topology changing, and then we get back to the same topology again. So it is going to be well described on this video. I hope that everyone can see this external video. Okay, this is the simulation. This is just the snapshot that I showed you. I want to take your attention to one point, that our spots are disappearing. By the way, this is actually a very slower motion, but I made it very fast, as you can see. You are gonna see it. In total, this video was like 50 minutes, something like that. I make it a bit faster. So anyway, we run the simulations and we observe that we have the spatiotemporal dynamic, the patterning in the dynamics. We see the spots patterning, and then they disappear and they come back again. It is time periodical solution, spatial and temporal, both. Yes, and like a heartbeat, you can think about it. Okay, from the L2 norm, this is one good thing that one actually proves our numerical methods really work well. That we can see the periodicity in our L2 norm graph. L2 norm graph shows you the time periodicity in the dynamics. That this belongs to the same simulation. I just showed you the video. And I told you that the other geometry, the non-convex geometry is our attention, takes our attention as well. So we do a similar analysis on the ring-shaped domain, again choosing the same rates of the diffusion, and it gives rise to same patterning. And just for a better understanding, I put the 3D view of it. So now you can see how in this video, it is also seen on Zoom, right? Is it seen on Zoom? Okay. In this video simulation, you see like mountains and valleys. This is not a Turing type of behavior, because in Turing type of behavior, the pattern stays same in the long term. But in this kind of behavior, where we choose the parameters from our Hopf bifurcation, transcritical type of bifurcation regions, it gives rise to these patterns. You might be wondering the parameters. Actually, I didn't put the parameters here, but I wrote them explicitly by making a nice table that for each simulation, which parameters that I use can be found out. One thing I can say about the choice of parameters, they're all done by the theorems. Because we want to show, like, support our findings. Okay. So this is now, we arrive at a conclusion. So what did I try to show you today? So parameter spaces is an important issue, because parameter choice is quite important in the numerical analysis part. And I showed you the Turing diffusion instability regions, as well as Hopf and transcritical type of bifurcation regions. And we have some nice conditions. They are derived for the pattern formation. It gives us relationship between the domain size, how large or how small the domain size should be, depending on the system parameters. And we do our finite element methods using the small perturbations around uniform steady state. And we use Newman boundary conditions, zero flux boundary conditions in our eigenvalue problem, of course, in the finite element simulations. And we have time-independent, like spatially inhomogeneous patterns, Turing type of patterns. And we have time-dependent, periodic spatially inhomogeneous patterns, spatiotemporal patterns, simply. And our two norms graphs are actually supporting our numerical observations. And now, with adding cross-diffusion to the system, we have now the ability to relax the Turing diffusion-driven instability condition. That we no longer need the long-range inhibition and short-range activation. Sorry. And so when we choose diffusion parameters equal to each other, as you can see in the simulation, I tried to mostly show the simulations where we have the equal rates of diffusions, which is not possible in a classical reaction diffusion system. And yeah. And one another thing I want to show you, that because I also mentioned it in the abstract, that I want to talk about it as well. So there is an important problem in our hands, that during the growth development, because of the hypoxia is defined as because of the oxygen deficiency in the center, starting from the center. Because actually tumor cells are actually following. There's one leader and follower. So we have a really reaction diffusion system. We might represent this as a reaction diffusion system. And we can do our analysis on annular domain actually resembles this kind of problem. So as the growth continues, it becomes the hypoxic stage that we have it. Like if we think about the cross-section of the tumor, we will see a hollow sphere. And so this is what I'm trying to understand now. What happens if the thickness of the annulus gets reduced to 0? Like what happens if we think about like a string, like a circle, basically. I try to understand now. This is what I'm working. So there are a few open problems that me and my collaborators are also working. Let me finish. Sorry. Yeah. What are we trying to do now? Stability analysis of the reaction diffusion system on growing domains, because all analysis here was done based on the stationary domains. In growing domains, think about our system will include time dependency in the reaction terms, which is making the dynamical analysis quite difficult. So we try to answer. Also, with linear cross-diffusion, with like different kind of reaction diffusion systems, we are trying to do that. Non-linear cross-diffusion has not been done. It is done. It's studied, but it's not well studied. Like we don't have much knowledge about non-linear cross-diffusion. Our cross-diffusion is linear in our case. And weakly nonlinear analysis might be a good approach other than the linear analysis that we did here. And multi-component reaction diffusion system, this is what I'm now working at the moment. Because our analysis was to understand the two-component system. We know that there are a lot of models including multi-components, like 2 activator, 1 inhibitor, 2 inhibitor, 1 activator, or more even. And so there is another thing I am working on. It's the bulk surface. Like all the analysis I showed you today was done inside the bulk. Now we want to understand like from bulk to surface. So bulk surface system that I'm trying to understand. So on the discretization on the right hand side is a finite element discretization. Somehow I try to understand the cell signaling cascade, like modeling it as cell cortex and cell cytosol. It's all like doing the analysis on geometry and trying to understand the relationship, the cell signaling between the extracellular matrix and inside the cell on a cellular level, inside the cell or outside of the cell. And this is also one of the things that I try to understand. What if we have the inner circle to be really closing to the outer circle? So what kind of analysis does this require? We would like to answer this question. And thanks to these people, this work has been partially carried out in Isaac Newton Institute in 2023, December. And here is the QR code for my published work. Thank you. These are my references, and I would be happy to answer your questions.